So it's a very long title, and I'm going to talk much more than microbiomes today. So my interest in human population genetics started when I learned that we can use human DNA to understand human genetic history, right? We are a very young species, but in the last less than 100,000 years, we have managed to move out of Africa and adapt to different parts of the world, going from cold climates to desert like in the Gulf. One thing at that time that was not resolved was how did humans populate the Pacific islands? 2020, and I hoped that I would one day get to work to resolve this missing piece of information. In 2021, my colleague completely trashed that dream when he published this paper looking at population histories of Oceanian populations. And this whole puzzle of how humans moved out of Africa has been very, very close to me since 2006. Now, Oceania has been resolved. The only place left was Hawaii, and I really wanted in. So I talked to Alex, the person who published this paper, and I found Mark, a wonderful capstone student from Fiji, here at NYU Abu Dhabi, and we started looking at human settlements and timing of human arrival in Hawaii. So in the plot on the top is a principal components analysis. Each dot there is an individual, and we're looking at genetic data. So the dots that are closer to one another indicate that these individuals are more closely related to one another. Dots that are far apart indicate individuals that are not closely related. In different colors, we show different continental populations. And you can see the spread of green dots going from Europe to Oceania and also to East Asia. So historically, that's what Hawaii is, right? They're Polynesians with recent admixture with Europeans and East Asians. At the bottom is what we call admixture plot. In the bottom, each individual is a column. It goes from 0 to 100, and different colors indicate different ancestry proportions. So if we look at the top plot, where I have all the labels written, Africa, America, East Asia, Europe, Papua New Guinea, and Oceania. These are about 1,000 individuals we used from previously published results or previously published data. And those individuals were used to train a model to dissect all these different ancestries, continental ancestries. At the bottom is Hawaii, right? So we can clearly see that in these 4,000 individuals from Hawaii, they have mostly yellow, meaning that their background comes somewhere from Oceania. They also have traces of green and red, green indicating ancestry from Europe, red indicating ancestry from East Asia. But if we really zoom into these plots, we also see traces, and this is in the inset, traces of Americas on the top, this kind of blue shade. So these two plots, these are the bread and butter of a population geneticist. These show that native Hawaiians are admixed between Europeans, East Asians, Polynesians, and potentially Native Americans. So what I showed you earlier is results from what we call global ancestry. Those analyses can tell us a little bit about what our different genetic ancestry components we have in our genome. But in the last 5 years, 10 years, population genetics has, people in population genetics have made magnificent developments in data analysis. So what we now have are tools that we call local ancestry tools. So these local ancestry tools allow us not only to ask what are the different ancestry components, but pinpoint in the genome where these components start and end. So in the plot on the top, the one that has mostly blue is a karyogram of the human genome, autosomes. The karyogram on the blue with lots of blue indicate that these are, this is one individual with a lot of Polynesian ancestry, more than 99%. In the middle is an individual that has 50% ancestry from Europe, shown in red, and 50% ancestry from Polynesia. The one on the right is a karyogram of individual that has ancestry from East Asia and Polynesia. Notice in all three plots, these are just representative plots. We have individuals throughout the spectrum. We see these green segments. And these green segments come from Native Americans. Right? So both admixture and this genomics result indicates that there is a Native American ancestry in the Hawaiians. So the next question we wanted to ask was where this Native American ancestry comes from and when. We figured out when part, where we're working on it. So at the bottom is a tracks plot. So if you look at the karyograms, you see that these segments that come from different ancestry have different lengths. Right? So in the middle, if you look at the 50% European, 50% Polynesian individual, everything in red is in one chromosome, everything from blue is in another chromosome. Right? So what that means is this is a first generation individual whose one parent was Polynesian, the other parent was European. On the East Asian plot though, it's all mixed, right? So what that means is this person had a parent who was a mixture of both. Right, so what we can now do, because that is the case because recombination can break down all these different track lengths and make them shorter over time. So we can now model these track lengths. Sure. So what we can do now is look at these track lengths. So the ones that are shorter, and look at the distribution of different longer and shorter track lengths, and we can model when these different ancestries integress. So here what Mark did was modeled Native American, East Asian, and Polynesian tracks. And with this tracks analysis, what he finds is that on a Polynesian background, European and East Asian component merged very recently, whereas the Native American component introgressed a while ago. He then looked at another subset of Hawaiians that have slightly different composition of these three ancestries, and he sees the same result. And then he looked at another subset where it was basically Polynesians only, and he sees the same result, right? So Native American ancestry came into the Polynesian background a long time ago. Now, using this information, we can do mathematical modeling to estimate precise time when the integration happened, and it turns out that integration happens around 870 years ago, or around 1125 AD, which is corroborated by several archaeological evidence. The next thing we want to do now is, okay, this is the genetic composition of Polynesians, but where did the Polynesians came from, right? So in order to do that, Mark looked at only the Polynesian component, and that's another beauty of local ancestry is instead of looking at every single ancestral component, we can respect the different times of admixture, and we can only select one of the components and ask what is the history of that particular component. And this admixture-based results, so Hawaii is here, the next closest population to Hawaii at modern times is Rapa Nui. We can trace it back to Tuamotus, right? So our model here is that humans in Polynesia expanded out of Samoa. They eventually reached the Tuamotus. This is the archipelago, and then from there, the migration into Hawaii happened. And this Native American admixture, where we have a line missing here, actually occurred in Tuamotus before the humans actually reached Hawaii. Can I get rid of, okay, all right. So with hard work of Mark, we have now a framework that we can use to look at population histories of humans elsewhere. Potentially, we can extend it to non-human species as well. So the next groups of humans that we wanted to look at was populations in Himalaya. And that's because I am actually from Nepal, so Himalayan populations are close to my heart, but also Himalayan populations are highly, highly, highly underrepresented in genomics literature. We know that India, even though India is surrounded by oceans on three sides and by mountains on the, throughout here, So this is Himalaya here, and then there's Hindu Kush Mountains here. It's surrounded by geographical barriers, but there have been many historical migrations that have happened into India. That the one that's super important is this ancient hunter-gatherers that entered India thousands of years ago, and somewhere from here they actually went into Australia and became, the Aborigines in Australia today are descendants of these ancient migrations, right? So lots of arrows here indicating that people moved around quite a bit. But what we don't understand is, so if we look at people from India and people from East Asia, they are genetically very distinct, indicating that there has been no gene flow and Himalaya has played a major barrier. Is that really the case? I don't think so, because I grew up in Nepal and I know, I've seen people that kind of look like, both. They have both features, right? So what I wanted to do was look at right here, because evolution always happens at the boundaries, right? So people who are here, they don't need to mix with East Asians. People living in mainland East Asia, they don't need to mix with populations here, but what's happening in these fringes? That was the question. So in 2016, me and my wife, who is now my colleague, we went and collected samples from 20 different ethnic groups, about 500 individuals. We collected saliva, height, feces, and whatnot. This is just one of the population. And I like to put this picture because this is from our work in the nomadic Raute population. Raute are one of the last, actually the last nomads in all of South and Southeast Asia. Actually, all of South Asia. So here again, we have a principal components analysis. East Asians are here in red, green, and cyan are individuals from South and Southeast Asia. These are individuals from India. And look at this. This is like Himalayan populations. We have high altitude and middle altitude populations here that are Tibeto-Burman or Sino-Tibetan speakers. We have a mix of Indo-European and Sino-Tibetan speakers here. These are primarily Indo-European speakers right next to the Indian population. We thought altitude would be the major barrier to gene flow, but actually longitude is better correlated with this climb. Again, we ran admixture. With admixture, we can look at how many different ancestral components there are. The best model was either K9 or K10. In K9, we have Africans here, Native Americans, Oceanians, Europeans, all of many Indian populations here, and these are the Himalayan populations. And this is Southeast Asia, and East Asia and Southeast Asia. Look at this, right? So there is a line of red indicating that there is this ancestry component that goes from high altitude, middle altitude to lower altitudes. But there's also this blue color that goes from, that is shared with South Asian populations that is present in many, many, many Himalayan populations. Now, Raute, remember the nomadic people of Nepal? They get their own component and we don't know what their ancestry is. So what we can now do is remove the Raute from the data, run admixture again, and project the Raute onto that data to say, okay, if we were to not have Raute in the data and rest of the components were present, what would the Raute genome or what would the Raute ancestry look like? And these two Raute populations are right here, and they also now show East and Southeast Asian admixture. And then if we go to K10, what we see is this Himalayan component, this East Asian component red, breaks down into cyan and, Dodger blue and red, indicating that there were two distinct genetic ancestries in Himalaya. So I'm the kind of person who's not satisfied by only one analysis. Then we did PCA, showed that Himalayan, at least the Nepali populations are admixed. We showed this, where we see both of these components at K10, K9. We ran another analysis called QP-ADM, in which we can actually mathematically assess the admixture proportions and assign a p-value to it. Everybody loves p-value. So this is the admixture proportions using QP-ADM in only the Himalayan populations. The proportions are similar, but one thing that I want to point is every single Himalayan population, regardless of where they live and what language they speak, they actually have South and East Asian components. So they're admixed. We also ran, we're in the process of running local ancestry. Here is a Sherpa individual that has mostly orange, which is an East Asian ancestry. A Chepang individual, Chepang is also another forager population, historically known for their foraging habits, but they have settled down now. We have Tharu individuals. Tharus are indigenous population to Tarai, and Tarai is like southern part of Nepal, and they are known to be resistant to malaria. And we have Chetri, who is basically someone who looks like me. Okay, so the Chetri, they have both East and South Asian. So South Asian here is blue, East Asian is orange, whereas the Sherpa have a little of blue, but they do have some blue nonetheless. So then the question we wanted to ask was, okay, so they have East Asian and South Asian ancestry, but where does the East Asian ancestry come from? Because there are also many, many, many East Asian populations in China. So this is a work in progress, but Mark did this mass MDS analysis in which we can again take only one of these components and plot it and do the MDS analysis, and it gives us two axes. And within this axis, we can ask, so each dot here is a centroid, like a, let's say like a median value for individuals in each of these different populations, and we can ask Himalayan populations which are here, which reference population they are close to. So a lot of the high altitude Himalayan populations are close to Tibetic populations, which is also corroborated by linguistic and cultural evidence. But then the middle hill population that showed the Dodger blue component, they are closest to Naxi and Yizu. So Naxi and Yizu are one of the two minority populations that are recognized in China, and they have been known to be historically residing in the Yunnan province, which is on the eastern tip of Himalaya, right? So it seems that the ancestors of the modern day Nepali populations came from Tibet, the Tibetic ones, from north to south. The middle hill populations, their ancestors moved westward from Yunnan. Another coolest thing that we found was the ancestry component, the yellow component that we found in Raute. We can also make this dendrogram to see when this component split, right? So the base here is Luya, which is an African population, and then we have the European population, Native American, and Raute split right after that. Papuan split right before that, and we have all of the East Asian populations here. So what this means is the Raute component could be an old component because it's the base of all of the East Asian component, which is fantastic because the Raute are nomadic. Linguistically, they fall at the bottom of the language tree, the Tibeto-Burman tree, and they are the last nomads remaining, right? So we may have discovered an old population without using any ancient DNA. And that's super exciting. So this is our model now for how Himalaya was populated. We think that there was north to south migration across Himalaya. There was east to west migration from Yunnan, and there were some South Asian population, ancient ones, that moved into Himalaya a long time ago as well. We're working on the South Asian part at the moment. So this is cool, but everybody wants to hear stories about adaptation, right? So what happened? Okay, these populations entered Himalaya. Something cool happened or not? So we're looking at adaptation. This work is being done. So all the population genetics of Himalaya, that work was done by Ishwar Muthukrishnan. He's a research scientist in the lab. And this adaptation is done by Rita Fami, who is also a capstone student under supervision of Ishwar. So Chepang is another population that used to be nomadic, but they're not nomadic anymore. Their diet consists of lots and lots of tubers, so they still eat, they forage, even though they have settled down, they forage. And these people hunt bats. This is a picture that I took when we went there in 2021. This guy just casually walked up to me and said, "Hey, sir, here's a gift for you. Here's a bat. You want me to cook it?" I was like, no. There's one thing that I will not eat, but the Chepangs eat it. And the concept of sanitation does not exist. So when we go there, when we went there in 2016, Yoshina and I, we went there. We washed our hands with a soap. We left the soap just out in the open. We also had a whole bunch of other things that was left out in the open. The only thing that disappeared was soap, right? So because there are no shops. There are the highest grade that's in the school is 6th grade. So people have no access to education. It's in one of the remotest part of Nepal, not at high altitudes, it's at middle altitude, but in a remote part. So, bats, and that's going to be important. So we then looked at the chipmunk genome and tried to identify signatures of selection in the chipmunks, and what do we find? The strongest hits are HLA, FGF2, and LCORL. There's also TLR4, and 5 minutes left? Okay. So TLR4, right? So these are genes in immune system. These are genes associated with height. So one of the things that really bothers me is when we look at non-European population, the best thing we can do is stop right here. Oh yeah, there's height done, right? But if this was a European population, there's a bunch of data sets out there that we can say, okay, where does this SNP map to? Where does this SNP map to, right? Because there's so much data out there. So what we're trying to do now is if we could take this data set, yes, some of the phenotypic data come from Europeans, but there are also phenotypic data coming from East Asians. Can we find some relevant phenotype? Are these SNP GWAS or PHWAS hits? Or in other words, have these SNP been associated with some phenotype in a previous publication? What did we find? So look at this. This is an LCORL signal. So this is permissive. This is the highest 100. This is the threshold, okay? So LCORL, there are a bunch of SNPs along this line. And there are about 32 SNPs. Green means these SNPs all 32 were associated with a bunch of different phenotypes. One of them was height. And green means the allele that was associated with taller height in an East Asian population in the PHWAS study, in the PHWAS database, was actually the minor allele in the chipmunks. What that means is this allele that is associated with, makes somebody taller or contributes to taller height, is not present at high frequencies in chipmunks. It's the other way around. So we measured height in chipmunks. This is in females, this is chipmunk, this is in males, this is chipmunk. So they are among the shortest populations. This data comes from a very small number of people because we were a team of 2 collecting these. But it actually is at least informative in the sense that, but when we actually went to the chipmunk villages, when I stand up, and I'm not a tall person, as you can see, I almost hit my head on their ceilings, right? So their houses are very small. So these are people who are short. So our working hypothesis is that chipmunks, being nomadic and hunting and gathering population, they may have, and living in an area that is not very productive, they may have been selected for shorter stature. And there are a bunch of other genes that I showed, the HLA and TLR4. We're super interested in functionally characterizing them to sort of see whether the signatures for selection is real and whether that gives them additional immunity against viruses, including COVID. And that's important because the Himalayan nectar bats, the one that I showed you, they are reservoirs for coronaviruses. So if you want to learn more about this, we have 3 posters from Mark, Ishwar, and Rita. Definitely check them out. I'm going to go quickly to microbiome. So one of the things that we have at NYUAD, and one of the reasons why I came to NYUAD is the UAE Healthy Future study. We have thousands of individuals from whom we have different types of data sets. We have we can generate genomic data, microbiome data, metabolomic data. And then we also have blood phenotypes. So we can actually go from characterization to functional, potentially translational. My capstone student, Sidania, did a lot of hard work to identify 2,700 individuals from the entire cohort. And from those 2,700 individuals, she actually was able to extract DNA. She was able to look at the survey and other phenotype data to identify a series of disease conditions that's prevalent in the UAE population. We selected the the highest prevalent ones, obesity and high cholesterol, and we looked at the microbiome composition. First, using 16S, we find there are Bacteroidetes, Firmicutes, and Proteobacteria that are dominant in the Emirati people, which is consistent with oral microbiome elsewhere as well. and this is something that Anik already presented. He also has a poster about this. When we look at association between disease phenotypes, like cholesterol and obesity in the oral microbiome, we did not find any signals for high cholesterol, but we did find signals for obesity in the 672 individuals. So when you have 672 individuals, you can break it down to a matched cohort, right? So that's how we picked the 602 anyway. So what we did was we looked at about 200 individuals. Some of them are obese, some of them are like normal BMI, and we see an even strong separation in the gut microbiome composition. They also differ in gut microbiome diversity, but then when we try to identify taxa or species that are different between obese and non-obese individuals, we sucked. It wasn't informative, just 1 minute. So what we did was we did metagenomics, and this work was led by Ahmed Shibol. He also has a poster on it, but when we do metagenomics, what we find is there are a bunch of functional pathways that differ between obese and non-obese individuals. We find a bunch of cool MAGs that are only present in Emirati people, and these MAGs are super interesting because right here, as you can see, this is a Petersky bacteria phylum. The purple is obese, orange is non-obese, and gray are reference. So there are, and each line here is a gene content. There are a bunch of MAGs from obese individuals that have these genes, not highly present in normal individuals, right? So we think that there are these bacteria. It's not only enough to say whether this bacteria is present or absent. We got to go down at the strain level and look at what are the differences in their gene contents. And this is an example where this bacteria is present in all individuals, but the content of their genome varies. So we are, since Shadi is here, he's supposed to be in sabbatical, but he's here. Thank you, Shadi, for coming to my presentation. We're doing metabolomics. So what we now know is we've characterized the microbe, we found association with obesity, we now have functional categories that differ between obese and non-obese individuals, but still it's a prediction right now, right? So we need Shadi's help to do metabolomics, and we can then say something like, these bacteria exist, they produce these chemicals do a bunch of things. And we also have blood marker data to see what are these bunch of things. That's it. There are these 3 posters. I apologize for the extra couple of minutes. And there are a laundry list of people to thank. And I am so grateful. And it's a privilege to be able to work with these samples and with these populations. And I thank everybody for giving me this chance. And I thank everybody in my lab for being part of this process. So this is who we are. We're a genetic heritage group. If you want to join our lab as a postdoc, research scientist, or capstone student, you can contact me. If you don't like me, but want to follow the updates from the lab, you can follow our lab on Twitter. This is who we are. And for the students only, this is who we really are.